So good day guys. Today I'm going to discuss about the Hymenolepis nana, also known as the Dwarf Tapeworm. So in this presentation, I will include the following: the morphology of the parasite, the biology and life cycle, its pathogenesis, the disease association and treatment, diagnosis, epidemiology, methods to hold reproduction or prevention, and I will also include some of point of interests about the parasite. So Hymenolepis nana is a cyclophilidian parasite that belongs to the family Hymenolepidae. Now, Hymenolepis nana is a parasite that causes hymenolepiasis. It's a very common tapeworm that can be found worldwide. And the group that is at most risk of this infection tends to be children. It is thought to be that their hygiene might be the cause. That's why the common infected with this Hymenolepis nana are the children. So its genus is Hymenolepis. Now, Hymenolepis species. There are many Hymenolepis species that has discovered by the authorities. There are roughly 400 species, and among those 400 species, there are only 2 species that is of interest parasite, namely the Hymenolepis nana and the diminuta. Now, Hymenolepis nana is a human parasite. Now, take note, it is a human parasite compared to diminuta, which infects the rats primarily. So, Hymenolepis nana is a cestode that infects the humans, and it is called dwarf tapeworm because it is the smallest cestode that infects man. Now, other names for Hymenolepis nana includes Rodentolapis nana, Vampirolepis nana, Hymenolepis fraternal, and Taenia nana. Now, from the word rodent, Rodentolapis nana, the word rodent, this name suggests that our parasite infects the rodents or the rats. So it can be found in rodents. It's also called Vampirolepis because compared to diminuta, Hymenolepis nana has hooks or equipped with hooks. That's why it's Vampirolepis. It uses hooks to attach to the lumen of the intestine. So from the word nanus, the nana was derived from the word nanus, which means dwarf. So this parasite is literally a small tapeworm. So other researchers would name it as a miniature tapeworm or a small tapeworm. So this is also its common name is the dwarf tapeworm. So take note of that. So Hymenolepis nana is the only known human tapeworm which can complete its life cycle in a single host. So unlike other species or other cestodes, they need at least 2 hosts to complete this life cycle. So it may need an insect intermediate host and a vertebrate final host. Now in this case, for our Hymenolepis nana, you just need 1 host to complete its life cycle, therefore improving its survival. So when we say it can complete its life cycle in a single host, it means that it does not require any intermediate host. So Diminuta needs an insect to complete its life cycle, while Nana does not. However, the most common intermediate host for our Hymenolepis nana tends to be also an insect, which is the beetle or the grain beetle. So man can harbor adult and metacestode stages. Now this metacestode is also other name for the larval stage. So some books would use the word juvenile stage or the metacestode stage or the larval stage. They are just the same. So don't be confused with those words. They just refer to the larval stage of the parasite. Now, brief history of our parasite. In 1851, Theodore Bilhars discovered H. nana in a biopsy of a boy. So recovered a countless number of Hymenolepis nana sa body sa bata. In 1852, von Siebold established that H. nana was a human parasite or is a human parasite. So it means that it primarily infects the humans rather than the rodents, unlike the Diminuta, which infects primarily the rodents. Now in 1887, it was discovered by Grassi that infections with H. nana does not require any intermediate host. So it has two alternations of its life cycle. It can infect an insect and then complete its life cycle, then proceed to a final host. And the other pathway is it directly infects the human. There are two alternations of its life cycle. Now those two alternations I will further discuss more in the life cycle section. In 1906, Stiles has identified an identical parasite with a rodent host and named it Hymenolepis fraterna in 1906. Now, let's go to the morphology of the parasite. Just like all other cestode, Hymenolepis nana follows the same process or morphology, which is the egg stage, the larval stage or the metacestode stage, and the adult stage. Now let's go first to the egg stage. Average size is 45 by 38 micrometers, so it's very small compared to Diminuta. It has hooklets. It is called the hexacan embryo. So there are 6 hooks or hooklets in the hexacan embryo. That's why it is hexacan. From the word hexa, 6, and can meaning the hooklets. That's why it's a hexacan embryo. There are 3 pairs of them. Polar thickenings is present just like the Diminuta, but unlike the Diminuta, it has polar filaments. So as you can see here, this is an image or illustration of an egg of a H. nana. You can see H. nana is a very delicate structure, has a delicate structure. Na siya outer membrane. This is the outer membrane, which is thin, a very thin membrane, and the inner membrane, which encloses the oncosphere. Now, the oncosphere is actually a larvae inside. So it is an oncosphere larvae enclosing it, which is enclosed with or by an inner membrane, which is also a thin membrane. And in this part is a polar thickenings. This is a polar thickenings Other name of polar thickenings is the polar knobs. It resembles on knobs pag makita nimo siya under the microscope. And on those knobs or filaments, in those thickenings, emerges the polar filaments or the hair-like structure sa H. nana. And this makes the H. nana different from diminuta because diminuta doesn't have filaments. So if you identify a Hymenolepis species under the microscope, para masayo na lang, you look for its filaments. If it has filaments, then it is H. nana. If it doesn't have, it may be diminuta. So there are 4 to 8 hair-like. So its filament consists of 4 to 8 hair-like structure, polar filaments. Its embryophore, colorless. So it is non-bile. No? Unlike the diminuta, it is bile, diba? Ayahang embryophore. That's why it's colorless because it's non-bile. So according to Belisario, he described it as clay color. So here is a typical image of the egg. So let's try to identify its parts. So the arrow points to its hooklets. There are 3 pairs of them. And here is the polar filaments which emerges from the polar thickening. Then the next stage is the cysticercoid larvae or the metacystal stage of the H. nana. Now cysticercoid larvae, same with the cysticercus of the Danish species. Now cysticercoid larvae and cysticercus is different. Now let's know why. Just like the cysticercus, it is a bladder-like structure. Its bladder, scientists called it the bladder-like structure or bladder because it resembles the bladder of the human. Now, unlike the cysticercoid has a well-formed scolex and not invaginated. So when I will illustrate the image of cysticercus, it's very different with the cysticercoid. Kaya naman god kay cysticercus, its scolex is inverted or it is fold inside out. So parang ganito. So the scolex of cysticercus is incised and inverted inside. The scolex is inverted inside. So this is the scolex. This is the scolex. This is the whole cyst. And it has hooklets. Or hooks. There are 3 pairs of them. So, para siyang inverted. And it is circular or spherical in shape. Unlike the cysticercoid, it has a well-formed scolex. So this is actually the scolex of the cysticercoid. And encysted, this is the whole cyst. This is the bladder. Okay. So in cysticercoid, it is not inverted, just unlike the cysticercus. And it has a tail-like caudal appendages. So it has a tail. So this is a tail. There are instances that H. nana cysticercoid has na siya 2 forms, which is a tailed cysticercoid and a tailless cysticercoid. Now, tailless cysticercoid is usually found inside a villus, where it kanang naga-develop pa siya into a cysticercoid larvae na tail. Other name of the cysticercoid larvae is the cercocystic cysticercoid or in short, cercocyst. Pero di man kay siya importante. So to be uniform, atong gamiton is this. The term is the schistocercoid larvae. Other feature of our schistocercoid, it is made of longitudinal fiber and spade-shaped with the rest of the worm still inside the cyst. So its calyx is inside the cyst. So here's the illustration of H. nana schistocercoid. So this is the cyst, the whole cyst, and this is the cyst cavity. So it's filled with fluid. And the sulot is the scolex. So as you can see, it is well-formed scolex. So it has a rostellium, suckers, and a neck. And around the scolex is the fibers. And this schistocercoid larvae is the infective stage of the indirect cycle of the H. nana. So actually there are two development of the H. nana. We have the indirect cycle and the direct cycle. So for the indirect cycle, the infective stage is the schistocercoid larvae. Now let's go to the adult worm. So the adult worm, same with other cestode, it has scolex, the strobila, and the neck. The neck is slender and long. And its scolex is subglobular in shape, and it has hold-fast organs. The hold-fast organs are the rostellium, the hooks, and the suckers. Now, there are 4 suckers, just like all other cestode. And it is cup-shaped. So take note, the shape of our H. nana suckers are cup-shaped, and there are 4. Other term for the 4 suckers is tetrad. Rostellium present. This is a rostellium, but it is short and retractable. So meaning it can adjust its rostellium. And the rostellium is equipped with hooks or armed with hooks. There are 20 to 30 hooks and just 1 row. The gravidoglottids, it has a size of twice as wide as long. So, compared to the tanya species, si gravidoglottids ni Hymenolepis nana is twice as wide. So, meaning para siyang rectangular in shape. Dili siya square. And its appearance is sac-like uterus filled with eggs. So, according to Belisario, when the mature segments or the mature proglottids becomes gravid, its ovary and the testes will disappear, and the uterus will hollows out, then being filled with eggs. So meaning sa gravid proglottids na di natin makikita yung testes and the ovary, only the uterus, and filled in ang iyang gravid with eggs. So in the next slides, I will show you to demonstrate a video to demonstrate kung anong itsura ng gravid proglottids na H. nana. So the mature segment, just like all other cestode, it has a complete set of male and female reproductive system. So for the male reproductive system, natay 3 testes. So there are 3 testes, and in between those 3 testes is a female genital, which is the ovary, and there is only 1 ovary for each cestode, for each proglottids. So this male and female reproductive system kay compose niya sa isa lang ka proglottids or segment. So isa ka segment is a 1 unit composed of 3 testes and 1 ovary. And these 3 testes, 1 on side of genital pore and 2 on the upper side. So, muna ang picture. This is an illustration of the arrangement sa system or reproductive system sa atong H. nana, mature proglottids. So, there are 3 testes. So, 1, 2, and 3. So, in between them is the ovary. Now, the one testes is found on one side of the genital pore. So, asa itong genital pore? Nasa lateral side. So, this is the genital pore. Here, this is the genital pore. And the pit sa genital pore is one testes. And on the other side, na wala ang genital pore, or on the upper side, are the two testes. So, mo siya. So, kung tanaw na to diri, so this is the testes, and there are three of them, right? So, these are the testes. And in between them is the ovary. And this is the vital area. And these are the genital pores. So the number of segments of our Hymenolepis nana adult worm is roughly 200. Or according to Belisario, it is in between 175 to 220 segments. So this is a video demonstrating the scolex of the aged nana. So as you can see, it has 4 suckers. So makita mo siya. It's cup shape. Right, cup shape. And it's a rostelium, very short, no? Very short. But it is retractable. So mutas, adjust niya. Taas. And it has hooks. It has hooks. The rostelium. Okay, the next video is the gravid proglottids. So as you can see, it is filled in with eggs. So wala kang makikitang testes and ovary in this proglottid kasi it's gravid. So nawala na yung uterus na lang natin. So as you can see, it releases an egg. Balik. So let's continue. So this is a picture of the comparison of the diminuta and the nana. So as you can see, diminuta is much larger. Ang egg niya is much larger compared to nana. But kay nana, na siya this hair-like structure or the polar filaments. So dali lang siya ma-differentiate with the diminuta because of its filaments. Now this picture is a histological section showing cysticercoid of H. nana developing the small intestine. So this is the cysticercoid larvae that is tailless na nag-penetrate sa bilos sa intestine. And then this is the cysticercoid and the adult. Other pictures of the H. nana morphology. This is the whole tapeworm, the scolex, the mature, the gravid. So this is a colonoscopy of a patient which has a case of hyperinfection of the parasite. So the parasite is able to, its ability to hyperinfect its human host. So this is because of the auto-infection. So our Hymenolepis nana is capable of auto-infection. That's why this patient acquire a hyperinfection. So this is the intermediate host of our Hymenolepis nana. So grain beetles. This is the grain beetle, and this is the flower beetle or the Tribolium confusum, and this is the mealworm beetle. Familiar, si mealworm larva. Ito yung larva na edible na kinakaon. So ang iyang adult stage is the intermediate host for the H. nana. So when you accidentally eaten a mealworm or a beetle na infected with Hymenolepis, you might get Hymenolepis. So, very uncommon lang na isa ka human makakaon ng beetle. Siguro nagtinanga lang siguro siya. Or wala naluto og tarong ang bugas. So let's go now to our biology and life cycle. So the biology or the life cycle of our H. nana has a dual pathway. So the direct and the indirect development. So let's go first with the direct cycle. Okay. For the direct cycle, it begins with the egg. Okay. The eggs was eaten, or when eaten by a host, it hatches in the duodenum of the small intestine, releasing the oncosphere larvae. So pag makaon ang egg, mudresyo siya sa small intestine, and then dito sa small intestine, specifically the duodenum, kay mag-hatch siya, releasing the oncosphere larvae. And then, this 6-hooked oncosphere penetrates a villus. So, matudayon siya sa villi sa atong small intestine. So, it mainly goes to the lymph vessels of the villus. So, atong villus or villi sa atong small intestine, natural lymph vessel. And ito mo penetrate ang atong oncosphere larvae. And then, it becomes a cysticercoid in 4 days. So, for 4 days, inside the villus, it will develop into a cysticercoid larvae. And after 4 to 5 days, the larvae will rupture the villi. So, mudako siya because nagdako na siya, it will rupture the villi. And then, it will re-emerge in the intestinal lumen. So, magawas siya sa villi, mag-re-emerge sa intestinal lumen, and then attaches its colex to the intestinal mucosa to develop into adults. So, once magawas na siya sa villi, it will re-emerge to the intestinal lumen, and then using its colex, it will attach to the intestinal mucosa. So, mag-attach na siya dito, and dito mag-develop na siya into adult worm. So, that is the direct cycle. So, the direct cycle does not include an intermediate host. So, it will just infect directly the human. So, that's why it's direct cycle. Kay direct ang infection sa human. Now, it takes 20 to 30 days from the time of ingestion for the eggs to appear in the feces. So, from the time of ingestion, makitaan daw ang eggs sa feces 20 to 30 days pa. So, eggs are passed in the stool. So, as you can observe, walay proglottids na makita sa egg, sa feces. It's because the proglottids undergoes degeneration. Now, what does it mean? Degeneration, kay unlike other species sa toad, si H. nana, very delicate ang kanyang structure. So, dali lang siya ma-disintegrate. So, inside the intestine pa na siya. Wala pa na siya sa gawas. So, inside the small intestine, pag mag-undergo siya apolysis yung proglottid, it will eventually or immediately disintegrate, releasing the eggs. So, that's why you cannot see any proglottids in the stool. Pwede, there possibility, makita ka proglottids, but it's very rare. Or not. So the eggs of H. nana are immediately infective. This is important feature of the egg of our H. nana. So, ang H. nana manggod, it releases thousands of eggs. Pero the possibility of them to survive is very low. So that's why it needs to reproduce more. And it is infective immediately. So improving its chances for infection. Yeah. And it cannot survive for more than 10 days. So kailangan within the 10 days na gap, kailangan maka-infect na siya human. And after 10 days, mamatay siya or ma-disintegrate sa external environment. Now let's go to the indirect cycle. So indirect cycle will include the insect or the beetle as the intermediate host. Now, it starts with the insect accidentally ingest a feces which is infected with the H. nana egg. So accidental ingestion of infected arthropod intermediate host like rice and flour beetles, and sometimes through fomites and water or food contaminated with the larvae. So si H. nana egg or the indirect cycle of the H. nana can use fomites. So example kay water and some objects na contaminated with the larva or with the egg or the larvae. And then eggs inside the insect will develop into cysticercoid larvae. And this cysticercoid larvae is the infective stage for the indirect cycle. So let's differentiate. For the direct cycle, ang infective stage is the egg. Okay. For the indirect cycle, ang infective stage is the what? The cysticercoid larvae. So remember that. Now inside the insect, they develop into infective cysticercoid larvae. So the cysticercoid larvae are released and will eventually. So nakaon na ni siya sa human or sa rat sa definitive host. So once makaon ang insect which is infected with cysticercoid, the cysticercoid larvae are released and will eventually develop into adult worm in the intestine of the host. So diretso na siya. Eggs are passed in a stool. So the same process ang i-undergo ni cysticercoid larvae. Pag ma-release siya dito sa small intestine, it will eventually attach to the intestinal lumen using its colex and then develop into adult worm. Now, let's recap. The definitive host of our H. nana is the humans. Also, sometimes rodents. But primarily, it's human. That's why it's considered as the most common tapeworm of man. So, the intermediate host is the optional. So, dili siya pilian. No? It may or may not use an intermediate host. So, depende kay H. nana egg. Kay tungod, si H. nana egg manggod is infective in 10 days lang. That's why di gyud siya magpili-pili. So it's na siya two alternations. It may use the direct cycle or the indirect cycle. So the infective stage is for the direct cycle is the embryonated egg or the egg. And the indirect is the cysticercoid larvae. Remember that. The site of localization, our adult worm resides in the small intestine. Namely the proximal ileum. Sa ileum siya. So the diagnostic stage is the egg. So wala kayong makitang proglottids in this stool because proglottids undergo degeneration immediately in the small intestine pa lang, sa sulod. So it will disintegrate. The lifespan of the adult worm mag-abot siya to 4 to 6 weeks. So, gamay lang yun kay siya. Chances are, when an adult host is infected with the H. nana, ang cause ng short lifespan is because of the immune system sa adult. So, self-limiting ang ating H. nana infection. So, malimit siya because of the immune response sa adult. So, motility is Chinese dragon-like movement. So, para siyang Chinese dragon. So, in the next slides, I will show you a video demonstrating the motility of the adult worm. The maturation from larvae to adult, it takes 15 to 20 days when they began to release infective egg. From larvae to adult, tapos mag-lay siya egg, 15 to 20 days to siya. So this is a video. So it moves like a Chinese dragon. No? Kung makita ka sa kwan, every time sa Chinese New Year na Chinese dragon, yan po ang motility Chinese dragon. Okay, next. Reproduction, just like all other cestode, they are hermaphroditic. Or other term for hermaphroditic is monoecious. Monoecious species are those species na ay complete set of both male and female. So, dili siya distinct. Dili sila separated, meaning nasa isa. That other is the dioecious. When you say dioecious species, these are the species na may distinct reproductive system. So, meaning separate ang ilahang male and the female reproductive system. So, example of dioecious kay ang Schistosoma species. They are dioecious. And most cestodes, they are monoecious. Nalang exemption for those cestodes that infects primarily the birds. So they are capable, if they are hermaphroditic, then they are capable of autocopulation. When we say autocopulation, it is capable of self-fertilization. And there are also cases that they can cross-fertilize. Those cross-fertilization, muna siyang kuan. The adult worm mag-mate siya with other adult worm. So mag-auto-fertilize sila. So because ako, if I am the tapeworm, tapos na ako nakita na other adult tapeworm, mag-cross-fertilize ko. So mag-release ko sperm, and the other tapeworm will accept my sperm. And then that other tapeworm also will release a sperm, and I will also accept its sperm para mag-cross-fertilize. So para siyang exchange, nag-exchange lang yung sperm. That's cross-fertilization. And cross-fertilization usually occurs when there are two or more adult worm inside the same host. And self-fertilization usually occurs when pag isa lang siya na adult worm inside the host. So it is capable of self-fertilize. And this self-fertilization occurs, mag-occur siya between the proglottids. So diba na may series of proglottids. So isa ka proglottid mag-release of sperm. And this sperm mag-wander around, tapos another proglottids will accept its sperm. Then that proglottid na nag-accept of sperm, mo to siya ma-fertilize. That is self-fertilization. Now, there are also cases na internal self-fertilization. So, meaning, siya mismo na fertilize. That same proglottid ma-fertilize niya yung sarili. Sperm. So, male organs develop before female organs. So, this is most common in hermaphroditic. So, I don't know why nanong mauna ang male organ before female organ. It may be that this is an adaptation sa cestode in which para ma-prevent ang self-fertilization. I don't know. Pero mo to akong nabasa. It prevents the self-fertilization. Proglottid development, just like other species, it takes 3 to 4 months. The mode of transmission is this: ingestion of eggs and auto-infection for the direct cycle. And for the indirect is the ingestion of infected arthropod. So take note of this. So auto-infection for this parasite is possible. So natay duwa, the external auto-infection and internal auto-infection. So external auto-infection happens pag ikaw infected with H. nana and then na-contaminate imong kamot kay tungod damak ka. Nagunit ka sa imong lubot tapos kaon ka, contaminated imong hands with the egg. So you auto-infect yourself. That is external infection, auto-infection. Naunay ka sa imong sarili. And this is common in high-risk groups and enables high worm load. So this is common in children. So magpataka sa lawg, gunis sa lubot and then magkaon. Ano siya, external auto-infection. The internal auto-infection happens internally. So this happens when there is a gut stasis or retroperistalsis. So internal auto-infection is when the eggs that is released by the proglottids mag-remain lang siya sa small intestine. So dili siya ma-passed out with the stool. Mabilin siya sa small intestine. And this is because of the retroperistalsis. Ang retroperistalsis is a reverse movement of the smooth tissue of the small intestine. And this is it is caused by irritation sa small intestine. That's why na-reverse or abnormal movement sa small intestine. And tungod sa retroperistalsis, mabilin ang egg. Kay tungod, di ba, reverse man. Dapat mong peristalsis pagawas. Pero pasulod man, so mabilin ang egg. And that egg, di ba, infective dayon siya immediately. And that egg will eventually hatch inside the small intestine and then continuing the same cycle, generating another generation of worms inside. And that may cause hyperinfection. Now, a quick recall, infective stage na H. nana. So, kamo na lang answer ani. So, you can pause for a while and then answer. Then na may answer po. This is the answer for number 1. Okay. Next. So let's go to the pathogenesis and clinical manifestation. So all symptoms of H. nana infection or Hymenolepis are generally produced because of patient's immunological response. So tungod lang siya sa response. Wala na siya laing like other parasite na mag-excrete siya enzyme to cause disease or to cause kanang infection. So tungod lang siya sa immune response sa host. Light worm burden is generally asymptomatic. So when you have less than 2,000 worms, you are considered as light worm burden. So asymptomatic usually ang light worm. And clinical manifestation for hymenolepis is headache, toxemia, dizziness, enteritis. These are just common symptoms sa mga parasite. And this is because of the immunological response sa atong one. This happens usually in the cases of heavy infection or heavy worm burden. Pag naganagid kayo parasites sa imong tiyan, this may cause a symptom. Toxemia is mo na siya ang the next slide I will show. So metabolic waste from the parasite can be absorbed and may cause toxemia. Ano siya? Diba, our cestode or H. nana doesn't have any digestive tract. So it mainly depends kung unsang available na nutrients sa atong small intestine. So when they ingest or digest your mag-produce mo siya metabolic waste. And this metabolic waste will be reabsorbed by your intestine and then go to your blood and then causes toxemia. And this is very dangerous. May cause poisoning sa internally sa atong body. And considerable irritation may also occur with large number of worms. So if you have a large number of worms, most likely karon kag irritation. And ang symptoms sa irritation or manifestations of irritation is the diarrhea or stomach upset, abdominal pain, and fatigue. So heavy infection may result in enteritis due to necrosis and desquamation of the intestinal epithelium cell. So necrosis and desquamation is caused by kanang migration ng mga parasites sa sulod nimo sa imong tiyan. That's why nagkaroon ka necrosis and desquamation. So, ang high-risk group na to are children. And children infected with this tapeworm will experience restless, irritable, and exhibit sleep disturbances. Rarely convulsion occur. So, muna siyang in the cases of heavy infection. Convulsion ay tungod po ni sa immunological response. So, ang atong tapeworm induces immunological response. So, humoral immunity. So, infection in children resolve spontaneously in adolescence. So, meaning self-limiting. So, if a children infected with the H. nana, pag mudako siya, eventually, mawala ang sakit. Mawala ang infection. Kaya tungod sa iyahang tungod kay ma-manage sa iyahang body or sa iyahang immune system ang parasite. So with time, regulatory immunity may limit or eventually clear the itch. So mo to siya, self-limiting. So experimental evidence has demonstrated that IgE or immunoglobulin E and the mast cells are involved in the expulsion of adult worms from the intestine. So ito yung responsible nga nung ma-self-limit ang atong H. nana infection. Si IgE and the mast cells. So according to Watanabe et al. 1994. So disease association is called Hymenolepis or Dwarf Tapeworm disease. So for heavy infection, mo siya, can cause enteritis and due to necrosis and desquamation. So treatment, the treatment of choice is praziquantel. This is a very common anti-helminthic na treatment or medicine. And other alternatives is the netazoxazide and niclosamide. Praziquantel is very effective na treatment for parasite. Now, you can consider yourself treated when pag imong stool are negative for H. nana eggs at 1 month post-treatment. So, kailangan treated na ang patient if mag-negative ang stool sample in 1 month post-treatment. So, kailangan magbalik ang patient sa imuhang after 1 month post-treatment. If nag-negative siya, then he or she is treated. If napa, then continue ang treatment. So, laboratory diagnosis. The method of choice is demonstration of the characteristic eggs in the patient's stool. So, meaning microscopy. So, you use microscopy to identify the egg of the H. nana egg. H. nana. So, wet prep is usually used. Wet prep. Flotation technique is also used. So, light infection, concentration of the stool specimen on alternative days. Alternative days is useful. Eggs are most easily seen on DFS or wet prep. Generally, proglottids are not recovered because they undergo degeneration prior to passage with the stool. So that's why di kay makita og proglottids in the stool. So another quiz. So kamo na po answer it. You can pause this video and then answer. This is the answer. Alright. Associated disease, hymenolepis, recommended drug, praziquantel, microscopy for diagnostic method. So epidemiology. Hymenolepis nana is found in areas with warm climate like southern USA, Latin America, and in the Philippines. So it is common in the Philippines. But nowadays, H. nana infection kay nag-lower. It's because of the hygiene of the people. So meaning nag-develop or na-improve ang hygiene. That's why wala na kayo cases of Hymenolepis nana infection in our region. There estimated 20 million people are infected with the Hymenolepis infection. So transmission, it's because of the poor sanitation of the people, overcrowding, and poor personal hygiene practices. So our disease is a institutional disease or familial disease. High risk group, persons residing in close quarters such as institutional settings, so it's an institutional infection, as well as children attending preschool or at daycare centers, according to Xavier. So direct contact plays an important role in the spread of this disease because eggs cannot survive long outside the house. So this disease can pass through from one person to another person. So importante yun ang hand washing as a prevention or preventive measure for this kind of parasite. So it is a familial and institutional infection. So when you treat a person infected with H. nana egg, it is important na kung kinsa pod ang household or kung kinsa ang members sa household, sila pod ay matreat because probably katong other members sa household nila may also be infected because it is a familial infection. It can pass from one person to another person, especially sa close good na setting. So methods to hold reproduction or preventive measures. Emphasis on proper hygiene. So this is important. Ang hand washing to break the chain of infection. And environmental sanitation. So, linis-linis. Infective cases should be treated thoroughly. So, mo ito ang ginaingon. Pag ikaw nakay-sakit na H. nana infection, might be katong close po nimo sa inyong balay, sa inyong institution, sila pod mag-treat because they might be infected also. Rodent control must be observed. So, food must be properly stored and protected from possible infestation with grain beetle. So, this infestation with grain beetle is very uncommon nowadays sa atong country because the storage of rice and flours kay nag-improve na. So, emphasizing the environmental sanitation or sanitation. So, point of interest. The eggs of H. nana is smaller than H. diminuta. Occasionally, epileptic seizures occur in infected children. So, epileptic seizures kay cause lang yapon sa immunological response sa body against the parasite. So, H. nana, the insect is always a beetle. So, pag panungkag H. nana or Hymenolepis, termitos, kailangan, yun, automatic beetle ang natong nana. Pero, in the case of H. nana, it is optional ang intermediate dose. Whereas in diminuta, kailangan yun na beetle to complete its life cycle. So humans and other animals become infected when they intentionally or unintentionally eat material contaminated by the insects. So H. nana infections are much more common than H. diminuta infections because it is spread by insects and spread directly from one person to person by eggs and the feces. Now, another nice to know facts. Si H. nana egg, when present in the feces, mag-release daw siya. Ang feces daw mag-release of volatile fragrant. So, volatile fragrant. So, what is the use of this volatile fragrant? This is especially for the beetle. So, si egg manggod mag-release siya when present in the stool, mag-release siya of volatile fragrant to attract or volatile attractant to attract the beetle to eat that specific species infected with the egg. So there are researches na nag-prove na nga naging may tabo. That's why improving the chances of infection. So si beetle makasimot atong fragrant or attractant, kaunon din niya to. So, nasalay setting, nasa research nila, i-compare nila ang feces na walay egg and the feces na infected with the egg. And most likely, the beetles mag-adto gyud sila sa infected feces. Now, when this beetle kay ma-ingest niya ang egg sa susulod na ang circular larvae. Now, when this beetle kay ma-infect og H. nana, kay si H. nana daw gina-alter niya ang behavior sa beetle or sa insect. So, it will alter the behavior, magkaroon siya ng surface seeking. So, ang pasabot sa surface seeking, kay mo magpa-expose gid siya sa gawas. So, increasing the chances of ingestion or of other predators or predation. So, makaon siya. There are chances na makaon gid siya sa rodent. It's very common in the diminuta. It's also the same with the nana. So, that's nice to know fact. Point of interest: in the Philippines, 2 independent surveys, WECO 1983, Cross-Etal, showed a prevalence of less than 1% in humans. So, infection among rodent was found to be low as well. So, this is because of the sanitation nowadays sa atong country na which nag-improve. This human tapeworm is also found among mice and less frequently among rats. Subspecies H. nana var. fraternal. So, mice, rats may be potential sources of infection. H. nana is the only, take note, only cestode capable of completing its life cycle without an intermediate dose. So, optional ang yang intermediate dose. Pila gani ang cycle? There are 2, which is the indirect and the direct cycle. So, H. nana is responsible for most tapeworm infections in humans with estimated 50 million to 75 million carriers worldwide. H. nana can also be transmitted through auto-infection without having to pass through the insect host. So, balik-balik na siya. Habitat, proximal. And that is the end of my presentation. Thank you for listening.